Yeah, apologies for the catchy title there. So DUS stands for distinctiveness, uniformity, and stability. And this is an example of a sort of practical application of machine learning. So this is a little bit of a niche area. It definitely felt like that was the case for me when I started working on this project. So new varieties must pass what's called the DUS test prior to marketing and to be protected by plant breeders' rights, which are ultimately IP for new plant varieties. Around 40 new candidate applications are received for spring barley per year at the testing centre. The testing centre for barley in the UK is NIAB, which is the National Testing Centre, and these are assessed for 29 DUS characteristics that show that they are distinct from all varieties held in the technical reference collection, which is at NIAB, and that consists of over 1,000 varieties. The minimum assessment period is 2 years, but often what we're finding is that a third year is required for this distinctiveness test, and that's becoming more and more frequent as the trait space in spring barley is shrinking. So just a little overview of the current pipeline, which is all based on phenotype data. So candidates are received at NIAB from breeders for the DUS assessment. These are then grown in a field season to get that phenotypic data back from those mainly morphological characteristics. And then the new candidate trait scores are compared to the technical reference collection to pick out subsets, which then need to go for further comparisons over typically, becoming more common, an extra 2 years, so the cycle can take 3 years. So ultimately, there is room for improvement in the approach. There's no genomic marker information really used at the moment. So we had some DEFRA funding to explore how to incorporate genetic marker information. Our proposal was to look at genetic marker-informed selection of relevant reference collection subsets in year 1 to try and reduce the maximum assessment duration from 3 to 2 years. This would allow for the sector to be more responsive and overall breeders can get new varieties out quicker, so it's a benefit to everyone. We aim to genotype the technical reference collection to explore correlation between the phenotype and the genetic space, and then to investigate if genomic prediction can be used for relevant reference collection subset selection. You might have to get your magnifying glass out for this, but in the sort of genomic informed approach that we're suggesting, the candidates would be received at NIAB, and immediately they'd be genotyped, and then we run genomic prediction to work out not just what the trait values are, but what the predicted subset comparison would be with that candidate. Those subset comparisons are then grown out immediately in year 1, and you can get to these comparisons earlier. So I should just say that I'm just talking about the distinctiveness test as part of the DUS cycle today, not uniformity and stability. So I won't spend too long on the genotyping aspect, although it did take a lot of the project. So we genotyped a proportion of the technical reference collection, which was 537 barley varieties. We teamed up with the James Hutton Institute, which isn't too far away from here, and combined data sets. They had 749 varieties as part of a previous BBSRC-funded project called ImPromote. That represented about 70% of the reference collection. And as you can see from the principal coordinate analysis there, it covered a broad range of the phenotypic diversity. So the traits. So this is where it gets a little bit more complicated. So the traits are sort of scored on a 1 to 9 basis. Some of them are a little bit more continuous in their nature, with almost a normal distribution. Some of them are binary, but it's really a mixture of sort of ordinal and nominal traits. We estimated narrow sense heritability, and this has also been identified in CJ's paper, which Wayne mentioned on the first day, shown there. And quite interestingly and strikingly, heritability was quite low for some of these physiological markers, and quite high, particularly for the traits with fewer categories. So the good news for us is that the genetic space is quite correlated with the phenotypic space. In this principal coordinate analysis here, we're looking at population structure estimated using genetic markers, population structure estimated using phenotypic data. The biological signal is largely the same. The clusters are separated by seasonal type and number of eero. When you estimate those distances in a slightly different way, looking at Manhattan distances, again, it's quite hard to see. Hopefully it's clearer up there. the correlation is fairly good, it's fairly strong. Even more so when you use min-max scaling on those phenotypes. And what this ultimately is doing is making sure the traits scored on a 1 to 9 basis don't have more representation in the Manhattan distance than those binary traits, which are the ones with actually high heritability. Binary traits, these are sort of ones like seasonal type and number of year row. So why did we want to use random forest genomic prediction? I think I'm one of the first people to talk about random forest in the conference. So random forest is a supervised machine learning algorithm that uses decision trees. I'm sure a lot of us have come across it before. It's powerful for different data types and has been shown to outperform common prediction methods, particularly for binary traits or categorical traits. And this powerful classification-based approach is why I wanted to explore as we're working with factors, really. It's quite computationally efficient if you're not doing it in a tuning process. So we're thinking about the long-term application of rolling this out to other testing centers. So the prediction approach, so each with 20 repetitions of tenfold validation. In the first approach, and we kind of use this as a sort of baseline comparison, treating the data as numeric. We estimated marker effects using RR-BLUP, and then in the kind of random forest classification-based approach, we use random forest SRC, the R package, and then treated the phenotype data as factors. We tested a couple of sort of flavors of random forest, one with just the default settings of the algorithm, and another with some inner-fold tuning of hyperparameters, and then more of a weighted major loci approach. So the hyperparameters that we explored were controlling the number of trees grown per forest, which is ultimately a tree represents a bootstrap of the sample data, controlling the mtri variable, which looks at how many molecular markers are selected or for contemplation at each node split or each decision in the tree, and then node size, which ultimately controls how deep the forest grows, how many cases are considered at the terminal nodes. The weighted major loci approach, we tested several methods for this, but in this scenario, we're reporting a kind of GWAS scan for each trait to identifying what the major QTL are, defining major QTL as anything with a minus log 10p value over 10. This is an example for ear row. You can see 2 major QTL. We then took the peak markers from those QTL and we increased the probability of them being selected in the mtri sample for each decision. And then once they'd been selected, we increased probability by giving them a higher split statistic, meaning that there's more chance that they'd actually be used for splitting. So these are some of the results, and here I'm showing prediction accuracy, so the test observed and predicted the correlation for each trait via the random forest approach, which is just with default settings at the moment, and the RR-BLUP approach. And this is correlated, prediction accuracy is correlated against trait heritability. You can see that the traits really do fall into these 2 categories of high heritability and low heritability. And interestingly, well, maybe not unexpectedly, random forest tended to perform better at higher heritability, whereas the RR-BLUP approach performed better at lower heritability. What's typically happening here is that those more continuous-based approaches, more continuous-based traits, RR-BLUP was better. In terms of looking at proportion of correctly classified calls, which is maybe more appropriate when we're working with factors, in this scenario, we'd rounded the RR-BLUP to the nearest integer, which isn't ideal, I appreciate, and then random forest generally performed better in that scenario. So these are some further examples of how we try to optimize the random forest approach. These box plots show RR-BLUP, standard approach, RR-BLUP with integer rounding, which adds more variability to the accuracies, as you can see. Random forest with default setting, and then random forest with tuning, random forest with weighting. I should have really done tuning and weighting combined, but I didn't get around to that. You can see in these 4 particularly nice examples, tuning and weighting did have an approach. That wasn't the case for, did have an effect. That wasn't the case for every trait, but typically where weighting was used, it always increased prediction accuracy. And these are traits all with high heritability, so this is where random forest was typically performing well. So once we'd established that the prediction was fairly accurate, we wanted to incorporate this into the actual testing pipelines. We are interested in predicting the values, but what we're really interested in is predicting the comparison subset to make sure that the predicted comparison subset aligns with the observed comparison subset. So in the current process, each candidate is compared to each variety in a technical reference collection, and is considered distinct if the difference in classes is equal to or greater than the class width for that character. These are some example class whips, and this is how the kind of distinctiveness approach, how some of these traits which are low heritability, more continuous, how some of that variability is accounted for before calling distinctiveness. So if distinctiveness is not shown across any of the characters for a certain comparison, that technical reference collection variety is grown alongside the candidate, and that's how these subset comparisons are formed. So what we're ultimately hoping to do is to predict what the subset comparison is going to be before looking at the observed data, and that's ultimately what we've tried to simulate here. So we took registered varieties from the last 10 years as candidates, and we simulated the subset selection using the observed versus the random forest predicted values. Interestingly, the predicted subsets were generally larger than the observed subsets. And you can see that here. What this box plot is showing is a point for each candidate, and then this is the observed subset, the number of individuals in the observed subset, and the number of individuals in the predicted subset. So the predicted subsets were generally larger, but on average, 50% of the genotypes selected for comparison in the observed approach were also identified in the predicted approach in that subset formation. So that's it's not bad, but it's not amazing. I think we can definitely do better than that. And this is just using some of those default approaches to random forest that I was mentioning. So I think we can take that further with weighting. There's also more we can do in terms of optimizing the overall prediction approach. The good news for us is this work has been, the funding has been extended for another 2.5 years. And we are going to genotype the remaining reference collection. We're going to further optimize the trait prediction, potentially using random forest, but also exploring some other methods or other methods of random forest, I hope. So we've explored a continuous approach, we've explored a categorical approach, and I think we should do an ordinal-based approach probably now. So that's probably the next step. And then also to validate some of these methods in side-by-side testing, so we can actually run the predicted informed approach alongside the observed approach in the field, and then ultimately rolling this out as a tool which people can use in other testing centres, other national testing centres, and other DUS assessments. So I just wanted to acknowledge my my colleagues at NIAB, our collaborators at the James Hutton Institute, the Imprimal Consortium for letting us use data, DEFRA for funding this work. And also, I don't really come from a machine learning background, so this has been a really useful resource for getting into this area, so I would recommend that to anyone. Thank you.